Hello, I'm Susan Rakes, Director of Learning at the Science Museum Group, and I'm delighted to welcome you all to tonight's open talk. Exploring issues around diversity and inclusion in STEM, the Open Talk series aims to shine a light on some of the less talked about stories in science, and to explore such themes as race and identity, unconscious bias in tech, and inequality in STEM. We've welcomed some fabulous speakers to this series, including Sir Tim Berners-Lee, Amanda Cozy-McQuarrie, MPs Chris Bryant and David Lammy, Dame Sally Davies and Joe Brand, while we've also collaborated with the likes of the Guilty Feminist, the Migration Museum and the European Space Agency. Our previous event in this series, Can Video Games Boost Your Mental Health?, brought together a brilliant panel of gaming and psychology experts to discuss how gaming may be able to enrich our lives, and is now available to view on the Science Museum's YouTube channel, as are all the previous talks in this series. But on tonight's open talk, what is sure to be a fascinating discussion, we'll focus on genetic databases, the banks of genetic data that enable scientists to analyse genetic diseases and to identify and develop new therapies and treatments. Tonight's panel will examine why a lack of racial diversity in these databases is a problem, as well as highlighting the efforts currently being undertaken to rectify this issue. We've assembled a fantastic panel to explore this subject, and I'm delighted now to hand over to our chair for this evening, a brilliant author, biologist and broadcaster, Aarathi Prasad. Aarathi, over to you.
Hello and good evening to you all. Thank you so much for joining us. My name is Dr. Aarathi Prasad, and I am delighted to welcome you to the latest edition of the Science Museum Group's Open Talk series. This is going to be a fascinating session. Ever since the discovery of DNA, the field of genetics has seen some of the most groundbreaking achievements in science: cloning, genome sequencing, important research into the prevention of disease, which has saved many, many lives to date. Yet, despite these milestones, genetic research has almost exclusively focused on people of European descent, taking up the vast majority of participants and patients, despite only representing approximately 16% of the global population. But what are the implications of this genetic bias? And what innovative research and analysis is being carried out in order to challenge this issue? To discuss this and more, please welcome with me our wonderful panel this evening. In no particular order, we have Professor David Van Heel. He's a professor of genetics at Queen Mary University, London, and the chief investigator at Genes and Health, which is a study working to analyse the genes of Bangladeshi and Pakistani people. We also welcome Dr. Janina Jeff. She's a geneticist and senior bioinformatics scientist with Illumina, and she's the host of In Those Genes, a podcast that uses genetics to uncover lost identities of African-descended Americans. And finally, we have Dr. Ananyo Choudhury. He's a senior scientist and reader for the Sydney Brenner Institute for Molecular Bioscience at the University of Witwatersrand. Welcome to you all. I'm going to start the discussion with Ananyo. Very good evening to you.
Very good evening, Aarthi.
Could you please, this is a broad question, but in a sort of potted background, could you give us a summarized background on what genetic research generally involves and what are the key goals it's trying to achieve?
Lovely, yeah. So I think that's a good question to start with. So as you mentioned, the reference human genome or the human genome. So what is the human genome? It's actually 24 extremely long strings of four alphabet, which is A, C, G, T, that represents the four building blocks of DNA. And so you have these extremely long strings of characters that make the genome. And if you add all of them, then you get up to 3 billion bases or 3 billion characters. So where we start is like we know that we share most of the genome, but then when we compare any individual to this reference genome, so we see that at certain positions of the string, this individual shows a different base or different character from what is in the reference genome. So these are what we call genetic variants or single nucleotide polymorphisms. And this is what is one of the, major sources of understanding diversity. So as we have sequenced tens or actually hundreds of thousands of genomes, now we know that any individual genome or any individual differs from the reference genome in about 3 to 6 million positions, or they have 3 to 6 million genetic variants, which is very tiny compared to the 3 billion base genome, which is like 0.1 to 0.2%. And despite this, we vary, and we vary a lot, not only in our physical traits, but also how we interact with the environment and pathogens, and also how we have different predisposition to different diseases. And so I think, and what we have seen in research over the years, a lot of these variations or differences have genetic underpinning. So I think our major focus is first to sequence genomes, to mine out new genetic variants, and then to investigate, setting up large studies, investigate how these variants are related to the traits we are interested in. So I'm kind of squeezing it, but this is probably the two major streams that genetics research is going in.
Can we look a bit more closely at genetic variants? So there's only a small percentage of them, but you said they can have a very disproportionately large impact. To what extent do these have an impact on health? And does the same variant have the same impact on all the different populations in the world? Or can that impact vary in people in different parts of the world?
Wow, that's another great question. So I just anticipated something like this. I have one slide. So if it's possible, we can just have that slide while I speak. That might help people to understand. So I think if we are looking into human traits, they can basically be classified into two categories. One are monogenic, which are really impacted or driven by a single variant. So it's like if you have that variant, you are going to have that trait. So sickle cell disease is a wonderful example where just having a single genetic variant could lead to abnormal shape of your blood cells and a lot of other problems that come with that. So these variants are generally valid for all populations you study in, and because of their strong impact, identifying the relationship between the variant and the trait is quite simple. But unfortunately, over many years of research, we have found just a few hundred such variants which have very strong impact. So that brings us to the second kind of variant, or traits, which are polygenic traits. So these traits are impacted by number of variants, where each variant impacts it slightly, but then taken together, they all make a difference to the trait. And in this case, for example, a good example is height. So a recent study showed that human height is contributed to by over 12,000 genetic variants. So, and each of them have some tiny impact. And for these variants, detecting impact is very difficult. And this impact is kind of modified by a lot of factors. For example, what other genes are present in your genome. So it's genetic background, which will vary between ethnicities, and the environment that you are living in, and the lifestyle you follow. So for identifying the impacts of these variants, they might just differ between populations from different parts of the world, but also even in the same part of the world, people living in different environmental conditions might show impact or not show impact. So the way of finding to be sure about what these variants do is to study them in different genetic backgrounds and different environmental conditions. And this perhaps kind of emphasizes the topic of today's discussion, that unless you have diversity in your databases, for these variants, you can only know so much, but you cannot go the full length.
You're based in South Africa, and you're conducting genomics research where you are in Africa. So how do these studies compare to similar studies on individuals from the populations that are more regularly studied from Europe or even Asia and the Americas? And where are the gaps? So which populations do we most urgently need more data from?
Wow, that's another great question. So maybe Luca, we could put up the next slide just to kind of show this. So this is one of my favorite slides, especially because I'm an African researcher. So here you can see that this is a study which compared genetic variants in 5 different ethnic backgrounds: Europe, East Asia, South and Central America, and Africa. And for each continent, there were 5 representative populations. And what you can see, Europe on the left and East Asia next to it, they have, and here each dot represents the number of genetic variants one individual has. And you can clearly see that Europe and East Asia has the lowest variation. South Asia, which is shown in like the rectangle, has slightly higher. And when you see Africa, which is on the rightmost side, shown within the circle, it has about 25% more genetic variant in each individual. And if you are wondering why would that be the case, so the answer lies in the history of our species. So maybe if we move to the next slide, we can look at that. So we all know humans evolved in Africa probably around 1 million years ago. And so this is a tree of all humans. So, and it starts from 500,000 years ago and come to present as you move down, and shows all the major divergences in the human lineage. So what you will see is that the first divergence in the human lineage, which is shown by the yellow arrow, is the divergence of San hunter-gatherers. Many of you probably don't know about them. These are groups who live in southern Africa, but they are the most diverged human lineage. The next group to diverge out were Central African foragers. We called them pygmy until some time ago, but we don't use that term because that's derogatory, but we call them Central African foragers. And then there was a split between East Africans and West Africans around 100,000 years ago. So if you are thinking where all the other non-Africans are, so you need to look at the right arrow. This points to a small outgroup of East Africans. And actually, a few thousand individuals from this group moved out of Africa around 60,000 years ago. And they formed, or they gave rise to all the non-Africans that we see in the world. So in terms of genetic diversity, if you are looking at diversity, that's the branch which you should not be looking at, because that's the most homogeneous and that's the least diverse. But somehow our genetic research has done the opposite. So we haven't looked, so African data sets are the least represented in genetic repositories, and we have been focusing only on that tiny outgroup. And that's why, to really understand more genetic diversity or what they do, you have to look at African data sets, probably some South Asian data sets. I think Prof. David will talk more about it. And then there are certain isolated populations which might be much more informative in terms of giving you more genetic variants and also helping us understand what they do.
Thank you so much. Maybe we can talk about this in the discussion later, but I'm also thinking, on the one hand, we need data from Africa and African populations. The flip side of that coin is to think about the benefits of that research going back to those people rather than just being used to inform in a kind of extractive way the research in the rest of the world. Well, thank you so much, Ananyo. That's been a very good introduction. I would like to move now to Janina Jeff. Good evening. Oh, it's early afternoon for you.
Yes, it is. How are you?
Thank you so much for joining us. I am, as well as being an acclaimed geneticist in your work in genetics, you're also a host of In Those Genes podcast, which is incredibly fascinating. You have topics of athletes and blackness, rhythm and blackness, fact and fiction, black don't crack. I love it. But could you tell us a bit more about the show, and how and why was it started? How are the topics chosen for the episodes, and what sort of things do you find it important to cover, or that listeners would find it important to listen to?
Yes. Can you hear me?
Yes, I can.
Yeah. So the podcast In Those Genes started I actually won an award. I applied for a grant at the time in 2018 by Spotify that was looking for women of color who were interested in podcasting, but who also had never done a podcast. And at the time, I had never done a podcast. I was only listening to one podcast. And so I applied, and out of 18,000 applications, I was selected as a winner of three. And that's kind of how the show started. To the show started as a what was supposed to be an entryway of teaching genetics to the Black community. And that really came about because whenever I would tell someone what I did for a living, I would get some common questions. I would always get, do you clone people? Which I'm sure everyone has probably on this call has probably gotten that question too. But then I would also get, at least from the Black community, questions about, well, what do you think about those genetic ancestry companies? And so I would always get lots of questions, and that really what led us to our first season, which is all about consumer genetic ancestry testing. And we really decided on that topic because we thought it would be an entryway to really talk to the Black community and the lay public about genetics. And so our first season focuses on every angle of direct to consumer genetic testing, the accuracy, looking at different testing, how does the test work, what are the data sharing components of it, a whole bunch of different topics. And then we also went in and started to we also went in and started to, in the second season, diving a little bit deeper into what we plan to. Can
So
you hear
just
me?
excuse me a minute, Janina. Ananyo, I think you're not
Yeah.
muted, so we can hear you in the background. So would you mind muting, please? Thank you. So, sorry, Janina, go ahead.
No, it's okay. And so our second season really started to dive deep in what we kind of see as the continuation of the show, which is taking each topic and really diving in and understanding what is fact and what is fiction. And that's mainly because a lot of the community seem to talk about genetics very casually. If we think about talking about it at our kitchen table, we're always, well, maybe you got this smile from mom or you did this thing from dad. And I think casually we love to blame genetics for pretty much everything when it comes to the uniqueness about us. And as we just heard earlier, genetics is much more complicated than that. There are a lot of different factors that contribute to who we are, and genetics is just one of them. And so we, in the second season, dive in a little bit more there. But we specifically wanted to focus on racial stereotypes because racial stereotypes have been unfairly blamed on genetics. And we think the lay public seem to really rely on the idea that our quote unquote less than 1% difference genetically can explain all of these differences that we have as individuals. And so we really try to dispel fact from fiction. And so in the second season, we specifically into traits and phenotypes that are also associated with racial stereotypes. And so we have an episode on the genetics of aging, which we call Black Don't Crack. We have an episode on the genetics of athleticism. We also have an episode on the genetics of rhythm. And so, and in the last episode, we talk a little bit about the genetics of body shape. And so these racial stereotypes and tropes, a lot of folks blame on genetics. And so we dive deep with experts in these fields, both from a sociology perspective and from scientific perspective, including my own scientific expertise, into really understanding what is real and what is not.
And speaking of racial stereotypes and race and sociology as science see it maybe differently, I wanted to ask you about conflating racial categories with geographic origins, because words are important in science, so are definitions. How do you see it? So what has the response been? Are people speaking about it differently, or how do you explain this?
Yeah. So I try to stray away from using the word race in our show and just in general because as we know, we're a very dynamic species. And so any type of categorization really minimizes and takes away a lot of information that helps explain who we are. And so I think it's really, really, really important how we talk about differences in groups of people. I think particularly in genetics, we should be thinking about it from a point of geography, since geography tends to explain a lot of the genetic variation we see within the human species. But I also, even within that, caution that we can't think about populations as these discrete groups. We should really be thinking about this as a continuum, as you can imagine, thinking about it much more on a quantitative basis, something almost similar as age, where there's this very unique number to each individual that describes their genetic background and their ancestral origins. And so I always like to make sure that we kind of steer away from categories and think about our genetic background as a continuum.
And so, to what extent would you say that what we're discussing today, the issue of genetic bias, say, extends beyond, let's not call it racial differences then, but geographic origin differences, to also contain a lack of diversity from varying geographic, social, and economic backgrounds in the data sets?
Yeah, that's a really good point. And in a lot of genetic research studies, people are starting to talk about this a bit more, right? What are the samples that we're collecting? How are we accessing those samples? Are we going through different types of communities, even within, like we know European ancestry is studied. In most of the genetic research, but are we also looking at socioeconomic status? Are we also looking at different extreme environmental populations? And to that extent, I think we still have a quite a long way to go. You have some really nice research initiatives like the All of Us program that is looking beyond racial and ethnic identity and really focusing on other things like looking at individuals who have disabilities, also looking at individuals who have different sexual orientation, in addition to different socioeconomic backgrounds. I will say that I do think it is a very difficult thing to understand. I tell people all the time when we think about who participates in genetic research studies, a lot of times this is a privileged population of people who have access to healthcare, who also have enough privilege to say, hey, I want to donate or be a part of research, where a lot of underserved communities have a lot of trust issues with participating in research study, rightfully so. And so I think, there is a huge gap there in beyond, like I said, racial and ethnic identities, thinking about what are some of the social determinants of health and what are some of these other categories that are also underrepresented. But like I said, the All of Us program has been just one of many that have been starting to explore and expand upon this idea of what we think diverse populations should be included in genetics research.
Thank you. And I think that I was thinking about the trust element because we saw that with COVID, with the pandemic. And I think that's why the podcast is so important, because you're connecting with communities and demystifying it and myth busting. Surely that helps to make people feel they can engage with it make it more accessible.
Yes, for sure.
Has anyone expressed the sentiment to you that, Ananyo was talking about how small the differences are, actually genetic differences, and but how that presents in the phenotype, in the way the variance that you see between people. But have you heard it being expressed that if we find genetic differences, it would lead to more of a divide in society based on the genetic data?
Um, that's a good question, and that's something I'm always fearful of, right? I think one thing we as geneticists do when we find something that could be explained by genetic background, we get really excited, right? We're scientists. We're like, oh my gosh, this amazing finding. And I always tell, when we explain this to the lay public, we also always have to reel it in and explain, well, how much of this given phenotype, trait, disease, how much does this one genetic variant or this region explain the entire disease, right? We talked about monogenic versus polygenic. And when we think about most of the common diseases are polygenic, right? And even find this nice admixture signal that indicates that there could be something there, we have to put into perspective, what does that mean? Um, are we accounting for potentially confounding variables like social determinants of health? Are we accounting for all the that contribute to a polygenic trait? So there is a huge fear that we identify these things, we could be creating a bigger divide. But that's why I encourage all of us, especially when we communicate this science, to really explain the full scope. Explain, okay, of this 1% that makes us different, um, this is a variant that we found to be associated with genetic background and given whatever else we're studying. This is how big or small it is in the grand scheme of things. It is an important finding, but we also need to talk about it that way. And then I think most importantly, we have to really change the narrative on how we talk about these differences. I encourage everyone, when we talk about these differences, not to always associate them with a disease or a phenotype that is somewhat detrimental. There are a lot of associations that we can make that are positive, like longevity. I think you see lot of a lot of papers coming out now talking about the genetics of longevity and just also understanding not just the genetics, but the environmental component. So thinking about how we can start to celebrate our differences and talk about them in a much more positive light will also kind of eliminate us using this to create biases and divide.
Thank you so much. It is often surprising to people that genetics is not as simple as you clone someone and you get a certain height or eye color. That it's much more complex, and it's not just genes but environment. And from what you're saying, some of that is histories. It's poverty. It's colonialism. It's all the impacts of what has come before. So thank you so much for your insights, Janina. We'll come back to you in the panel shortly, but right now I'd like to move to David. David, you're the Chief Investigator at Genes and Health, and that's a East London study. So could you tell us a bit about its mission, so how it came about, and why is it essential for the study of genomics in the UK?
Hello, Aarathi, and hello to our listeners. Salam alaikum to our research volunteers and communities. So Genes and Health is a study aimed at improving health in British Bangladeshi and British Pakistani people and communities worldwide, really. So I work in Whitechapel. I'm a doctor and a professor of genetics, and actually we see really easily locally lots of health inequality in British, Bangladeshi and Pakistani people and deprivation. It's right on our doorstep. These are our local ethnic minorities in East London. There's very high rates of diabetes, coronary artery disease, which occurs early. During COVID, the Office for National Statistics produced some studies, and British Bangladeshi people were 5 times more likely to die of COVID than white European UK people, British people. So there's a lot of deprivation. Also, there've been some big studies. You know, the UK has led with some big studies. People probably heard of UK Biobank, which was a study of 500,000 people, which has been fantastic. It's been absolutely outstanding science coming from UK Biobank. But UK Biobank is almost all healthier, wealthy, white British people. And actually, there's only about 250 British Bangladeshi people in the 500,000 of UK Biobank. There was very poor participation. So we wanted to do something about that. We've recruited 60,000 British, Bangladeshi and Pakistani people so far from Bradford, from East London, right across London, from Manchester, and we're expanding into West Midlands and throughout the UK. And we're linking, we've sequenced about 50,000 people so far. We're about to release that data. And the study is what it sort of says on the label. It's connecting genes to your healthcare. And in the UK, we've also got the National Health Service, which has fantastic longitudinal health data and health records. So we're connecting the two, and then we're also able to recall certain people for further research studies if there's researchers have other interesting things that they would like to look at with an additional consent step.
Could you say a little bit more about how the studies are carried out?
Yeah, so first of all, we explain the study to people and get informed consent from people to take part. Taking part is pretty easy. It's a spit sample in a tube and a short questionnaire and consent, so you can take part in 10 or 15 minutes if you want to. And we've found what works for British, Bangladeshi and Pakistani communities is trust, and you've got to do this face-to-face, often in an NHS setting, such as a primary care GP surgery, or in a hospital, or in a mosque, or in a community setting, a library, or a community centre. And actually, we found that writing to people, or text messages, or WhatsApp or Facebook just does not work for our local populations. You've got to do it face to face. If you've got someone who speaks, in our case, Sylheti or Urdu, even better. And people want an explanation, and they want a one-to-one explanation. They don't want a online sign-up, I'm afraid.
And how you must get similar questions to what we're discussing today from your community. So how do you explain why this is important to them? Is it by talking about the kind of diseases that we all from Asian backgrounds know, the Asian curse of heart disease and diabetes?
Yeah.
How do you find that common ground?
Yes, so there's actually a lot of interest in improving health and recognition that rates of diabetes, heart disease and so on are increased, and COVID has just amplified the health inequalities. We have a community advisory group actually led by Councillor Arsan Khan, who was our first participant from Waltham Forest, and our community advisory group has set our priorities as diabetes, heart disease and mental health. Actually, there's a lot of mental ill health amongst our local communities. So we research those areas. We actually bring researchers into Genes and Health. I don't know very much about some of these diseases. I'm a gastroenterologist, in fact, but we've got 120 groups of researchers working with Genes and Health now. And the joy of it is that people come with fantastic ideas that I haven't thought of, but we have a research database and facility that enables people to bring fantastic science to health deprived communities, which is what we were set out to do.
So we see the importance of gathering data where there is none, and from communities where there is none. So how do you feel about there are some researchers who advocate for, I don't know what to call it, so race-based medicine or understanding genetic variations-based medicine in the face of racial bias, not only in genetic databases, but in medicine overall.
So just in medicine, I'm a gastroenterologist, and when you look in someone's stomach or in their colon, it all looks the same. You can't tell what race someone is. And actually, medicine, when you're looking at an MRI scan or something like that, the investigations and taking the history from the patient is a similar practice, whatever your race. Well, actually, that's not quite true for treatments. And as well as bias in who takes part in genetic studies, there's quite a bias in who takes part in clinical trials and clinical trials of new medicines and testing of medicines. And again, quite a lot of white people take part in clinical trials, and they're just not tested on other racial groups. So we've just had a study of a medicine for heart attack called clopidogrel, and clopidogrel is a drug that it's called a prodrug. It needs to be metabolized to make it work. And people come in slow metabolizers and fast metabolizers. And if you can metabolize it, that's great. Clopidogrel will work for you and will stop your coronary arteries blocking up. But we've found that in Bangladeshi and Pakistani people, there's some genetic variants in one specific gene that slow the rates of metabolism down, and actually clopidogrel does not work for these people. And so we've seen in people, it's about half of the Bangladeshi and Pakistani population who are slower intermediate metabolizers. And if you don't activate the gene, actually you've got a 3 times chance of having another heart attack compared to people who do activate the gene. So we need to use, I think actually it's mostly going to be in drug treatments that a lot of use of genetic information should come in, and that does vary, as in this example, across populations.
I have the sense that for better or for worse, medicine has tended to be a one-size-fits-all sort of solution, and what you're describing is looking more in a more nuanced way, right?
Yeah, I mean, we all take 2 paracetamol tablets, right? Whereas you might need 1 and I might need 4. We don't do much precision medicine, as this is sometimes called, and particularly in drug treatments, one size does not fit all.
Thank you so much, David, for sharing your study with us. I would like to open some questions up now to the whole panel. I may direct it to one person or the other, though please feel free to jump in and share your thoughts. So one of the questions we had is that there are some countries that, I don't know if the biobank is one example of this, although you were saying that even though it's open to everyone, technically it does have a limited uptake. But there are some countries such as Denmark, Japan, and Korea, South Korea, that are working on creating country-specific reference genomes. Of course, I'm also minded that there is diversity within countries, so it wouldn't all be looking at the same populations. And they catalogue certain genetic variants that might contribute to health problems that their, I hope, diverse populations face. How far will this go to improve the data on genetic diversity, and then maybe, David, for you, the medical care? And that, as I was thinking about before when Ananyo was talking about African studies, that it should impact all people, no matter where the data is gathered. David, do you have any thoughts on that, firstly?
Yeah, so I think there are actually, pleasingly, increasing numbers of studies of diverse populations in genetics. It's not just what might be called a reference genome, which is what's usually used in sequencing to work out where each little bit of DNA has come from. Actually, probably what's more, we've already discussed in the introduction, that 99% of our genome is the same between us, and it's only little bits that vary. What's more important is to sequence lots and lots of people, and lots of people from different diverse ethnic backgrounds. And certainly that's what we've been doing for British, Bangladeshi and Pakistani people. And there are, there's big efforts in Africa. There's efforts in Australian and Pacific Islander populations, a study called Our DNA. There's some country specific. There's in Finland, for example, there's a big study called FinGen. Finland is interesting because it had a recent population bottleneck. So the population has grown from a very small community, which gives other bits of interesting genetics. We just need lots and lots of diverse studies, and then we can understand the entirety of human genetic variation.
Ananyo, do you want to add anything specifically referring to your work in Africa?
Yes, so I think, we are definitely lagging. It's still like, I think 95% or 93% of genetic data is still European. And so there's still a huge gap, but efforts are coming up, as Prof mentioned. And in Africa, we had something called Human Heredity and Health in Africa, or H3 Africa. There are initiatives in other parts as well. So yeah, the scenario is changing, but perhaps quite slowly.
And Janina, I wonder if you could say something about when do we get to the point where this precision medicine that David and we were all talking about, that precision medicine, which tailors treatments based upon a person's genetic data, but also lifestyle, as your podcast discusses, environment, physiology. How do we get to the point where that becomes easily available or accessible, and to many people?
Yeah, I mean, I think we still have a long, long way to go. I think that's just mainly because we don't fully know all of the data that we need to be testing and analyzing. But I think that there's a lot of work that's happening, and I think that we're getting, as technology evolves too, we're getting much more creative in the type of genetic information that we're able to provide and test, as well as the type of environmental data we are collecting. You see a lot of studies now that are collecting activity on Fitbits and Apple Watches. And as data grows, we're collecting more of it to really understand what are the different components beyond the genetics. But I think that is a very complicated, very complicated question. And so I think we'll continue to be at this is the state of precision medicine today, right? And that will evolve as the data sets grow, as we do more genetic studies in different populations, but also as we start to collect more data beyond genetics.
And David, in terms of yes, go ahead. I was going to ask you in terms of the work you've been doing, I
was going to add a little technicality for those who are interested. The bits that vary in the genome are really divided into two groups. There's a common set of variants, which are high frequency in the population. So, maybe 1 in 100 people have them. And actually, those are usually seen across most ethnic groups. There's the set of common high frequency variants are common to us all, broadly speaking. It's the rarer variants that often have a higher impact individually, that differ between each ethnic group. So, and actually, variants in white British versus British Bangladeshi or black British, the set of rare variants is completely different. And the rare variants have a lot of value and give a lot of information. And that's really the reason why we need to do lots of sequencing studies in different genetic groups.
That's a very important nuance. It's very interesting. Yeah. Thank you. So, Ananyo, I wonder if you could tackle this one to start. Humans. You showed us the population movement out of Africa and talked about when that happened and showed us the small numbers that went in different directions. But considering that humans have been interacting with each other for tens of thousands of years more, is the human genome simply too diverse and complex to get to a point where we can have, and I mean individual, precision medicine focusing on individuals' genetic makeup? It sounds like something from, science fiction, that there'll be something that can respond to our issues.
Yeah, that's actually a quick question. I think, the problem is, I think when we sequence the human genome, I think if you read the newspapers, we thought that we would have solved almost all the health challenges and we'll have all the drugs in 5 years. But I think what we have realized in the last 20 years or so is that these systems are incredibly complex. So, and the complexity of biological systems is something which we have just started scratching its surface. So, I really don't think that it's not possible, but I think there are certain new things that needs to come as innovations. For example, I think Janina mentioned that we need to go to other omics. So we're only looking at the genes, but throughout your life, your RNA profile changes much faster, your protein profiles change. So probably that could come in. And we have a lot of microbes living inside us, which is our microbiota. And they impact us in many more ways and more strongly than we ever thought. So studies are showing those. And perhaps integrating them would help. And also, I think the math or the stat we are using was designed or was thought up for much simpler systems where you have 1, 2, or 3 variables. Here we are dealing with systems which have hundreds of variables. So that's why we are trying to innovate more of data science, artificial intelligence, those kind of things to really make better sense of these data sets. So yeah, I think, we are making progress, and it's still a science fiction, but maybe with a few major breakthroughs. And for many traits, we might have something very close to precision medicine, maybe in a decade or so.
We've got little patches of precision medicine emerging. So there's a medicine for HIV called abacavir, and you've got to have a genetic test if you take that medicine to make sure you don't have a variant that will give you a bad side effect. Okay, so the FDA and again in the UK insists that you have the genetic test before you start the medicine. So that's a bit of precision medicine. But then also in the UK, once you get to my age, unfortunately, you get scored for your risk of heart disease. There's a thing called a Q risk score, which is based on age and whether you smoke and what your family history is and so on. And it doesn't take into account genetics at all. And so that's not really fantastic. And we've identified through genetics a small number of people who are at very high risk of heart disease. They've got a condition where their cholesterol in the blood is very high and has been very high for lifelong. It's called familial hypercholesterolemia. We've treated 50 people with that, none of whom knew they had the condition, and it's easily treated with statins and stopping smoking and so on. So there are bits of precision medicine, little patches, I think I'd call it, but it's not mainstream by any way.
Very interesting. Janina, I'm going to come to you with this one just because of your... sorry, Ananyo, did you have something to add?
Yeah, I just wanted to add another example where we are really kind of seeing precision medicine at work, which is, transfusion. So we are bringing in arrays which would allow you to identify the precise match for you, or the people who you should not be donating blood to you. So that's kind of would perhaps make the transfusion medicine much more precise and much more targeted to your genetic makeup. So yeah, just to kind of reiterate the point that we are having those patches where precision medicine can be seen in action.
100 years ago, we didn't have transfusion medicine, and we had no precision at all. So, blood groups are genetic, right? So, yeah.
It's great to have those examples. It's to see how far we've already come and what's coming. I wanted to ask Janina this one because of your podcast and your engagement with people who may not engage with genetics otherwise, on their own terms, in their own language. There are so many people around the world with limited access to things, lots of things, the internet, modern medicine, etc. How do we, but is it important? Do you think it's important to get to keep making genomics accessible, to make people comfortable, to get people to feel comfortable with engaging with genomic medicine?
Yeah, no, for sure. And our podcast is just one example on how to do that. And like I said, we focus on teaching genetics in a culturally attuned way. But as you might imagine, there are several thousands of cultures across the world. And so there needs to be an In Those Genes podcast for each and every one of them, or some form or fashion. But yeah, I agree. And we encounter this. In fact, at one point in our first season, we didn't realize, but a part of our audience was a group of students who were receiving their bachelor's degree while incarcerated here in the US. And so they did not have access to listening to the podcast. They actually read the transcripts from the podcast. And so I do think that access is a huge part. Access is all connected, right? When we think about how do we improve precision medicine, how do we improve, representation, it's also about access, it's also about transparency, and it's also about communication. The more we increase genomic literacy in communities throughout the world, the more engaged those communities will be with research, the more that we die, and that leads to diversifying the genomic workforce, that leads to diversifying data sets, that leads to diversifying the types of questions we ask, right? And so there is a huge return that's not just focused or not just limited to the scientists or the research, right? This is a big circle. And as we increase access in literacy, we also increase access in all of the different parts that really help diversify genomic research as a whole.
Those are really good points. It's thinking about the research agenda, who directs it, the funders, and who gets to ask the questions, and how to diversify. That's very, very important point. David and Ananyo, do you engage? I mean, David, obviously you do. You work with local communities. Do you have any points to make on the importance of making this accessible?
Yeah, we have a very active community advisory board who we consult on everything, and when we have an application for a researcher to use genes and health, if it's in a sensitive area like mental health or sexual health, we'll go direct to the community advisory board to say, is this appropriate? Is it a good thing to do? And for mental health, actually, I didn't know, but to our surprise, we found that there was a huge desire to look into mental health in British, Bangladeshi and Pakistani communities, but actually lots of things we hadn't thought about. Pregnancy-related mental health was had particularly come up. A number of other areas, and so you've got to do this. You've got to do research together, and us and them type research just does not work, and that's been really a core part of Genes and Health.
Yeah, I think I would kind of. Sorry, go ahead. Sorry.
Yeah, and unfortunately, in some parts of the world, there's been a historic culture of us and them type research, or we're doing the research on you, and that's not been a helpful background. But I think we're certainly, for our study, it's very much collaborative.
Go ahead, Ananyo. Yeah, I think a lot of the history of science and research was on other populations, which benefited one but not the others. Go ahead, Ananyo.
Yeah, so I think, as a research group, we have also worked in several rural settings in African countries where they have more pressing challenges compared to understanding or knowing genetics. But what we have seen is that despite these people are more altruistic than you would think they are. So if you really kind of tell them that this might not benefit you, but this might benefit future generations, they often are very interested in participating. So that's an observation that we have had here. So it's not that you have to convince them a lot or they're always scared. And the other bit is that, what we have seen is, for example, we have a cohort that's running for 10 years. So we went them once and we went them after 5 years, and we still had many of them come back. So it's more like they really, if you really kind of build long-term partnership, they stay with you, and so it's up to us that how do we engage them, and or are we interested in building a relationship or just taking the samples and running away. If they see you are honest, they often reciprocate back with support. And the other aspect perhaps here, which is I just wanted to add, was that we haven't talked about it, but there is something which we are talking, I think Prof. David kind of touched upon it, is called precision public health. So here you are not looking at precision medicine at an individual level, but at a communities level. So, there are communities living, and some communities have more requirement of particular medication, devices, hospital care. So especially in settings where resources are limited, if you can really still build models of where the requirements are based on genetic and epidemiological data, that might really affect big change in terms of how efficiently healthcare can reach people. So, there are indirect benefits to the communities, even if not at the individual level.
That's so interesting. I saw on your podcast website one of the programs, Janina. It said something about it takes a village to raise it takes a village, and a Somaliland friend of mine said that to me the other day as well, and they very much live like that. And that the idea of a communalistic society where, the benefit, it's really interesting, the benefit may not be for me, but it's public health and it's for people.
That's a really powerful message, and helping future generations, and that's a big reason why people take part. Yeah, and also we put out the message that if you want to do something, you've got to take part in the research studies, and whether it's a genetics and health study or a clinical trial, this is how we make progress in medicine. And if all the white British people keep taking part and other diverse groups don't take part, and diverse can be ethnicity, but it can also be male and female equal, it can be equal ages taking part. We need diversity in our research studies. They'll be better.
On that point, when the human genome was fully sequenced back in the early part of the millennium, 2003, I think it was, do we call it the human genome or do we call it the European genome? How much input has there been in that? What would you say?
Yeah, maybe I can take a start. Yeah. So I think, the idea was to still make it diverse. I think they started with a pool of 20 participants. They had and represent, I think, one or more representative Africans and East Asians. But somehow it happened that the final genome was was based on only 8 individuals, and the way the technique worked, there 66% of this was from a single individual. So this was more like, more closer to a European genome. But I think, we have to really say that in the last 20 years or so, we have developed genome build. So we started with the genome build 1, and we are currently at build 19, and some of these gaps have been bridged. And I think there is one initiative which is called the Human Pangenome, where you are trying to really do not a single reference, but look at the reference also as like a collage of genomes, so that you can align, if you have different populations, you can align it to them. So these initiatives will perhaps, make this reference genome the true reference genome in like a few years.
That's so interesting, because it reminds me of Linnaeus, who classified things, classifying himself as the type specimen of humans. It's a Swedish man.
The human genome has done a very good job of being a reference sequence, that because 99.9% of everybody is the same, right? So those bits will work, whoever was in the human genome, and so it's a very, very good scaffold. What we haven't done is sequence lots and lots of different people and apply them to that reference sequence, and I think that's probably more important. Every ethnicity, every ethnic subgroup. We've done some studies of British Pakistanis in Bradford, and actually found 11 different subgroups amongst British Pakistani people. So you can dive even deeper. So we should be sequencing all those subgroups as well, right? And I suspect there's subgroups within subgroups, but I won't go there.
Thank you. So is it feasible, do you think, to attempt to sequence the genomes of every, I don't know if you'd call it ethnicity or population, as opposed to, think of humanity overall? Could this even be achieved?
So there's a big push by life sciences industry to do it. So there's many life sciences companies want to know the differences in rare variants. Rare variants are very informative for drug development. There's one company in America who sequenced the coding bits of the genome of about 2 million people. So I think it is happening. Not all in academic open settings, but it's happening.
Janina?
No, I couldn't agree more. Is it feasible? Or do I think we will have genetic information on every single person? Would that be the norm? Um, I think we still have a long way to go, but I definitely think it is feasible. The reason why I'm asking, I'm trying to think of what is one thing we have on every single individual in the world that is like collected, and I can't think of one yet. Thinking about genetics being one, but there are a lot of large genetic studies, millions and millions of people in the UK and the US and Asia that have all been taking place. But I mean, we're talking about 6 billion versus a few million. So I don't know. I guess to answer your question is, do we need to have all 6 billion? Do we, what does full representation look like? Which it may not look like all 6 billion, because we know that we're all related. And so we probably don't need that much. But yeah, I think that's definitely something of interest for the reasons that David had already mentioned.
You were saying earlier when you were speaking about your podcast that the people you come into contact with, members of the public discussing genetics, do go out and have themselves tested on these, the tests that you purchase online. My sense is that it's something that people are at least fluid with or curious about. Is that your impression too? I mean, maybe that helps to, I wouldn't say normalize, but it gives it a familiarity.
Yeah, I mean, perception of genetics and engaging with consumer genetics or in participating in genetics research studies is very different from different regions and different communities across the world. So it really does vary. Consumer genetics company has been an entryway of getting a lot of people, I mean, massive amounts of people involved in genetic research studies. 23andMe is a great example of that, where they have data sets of well over 10 people. And so I think that is definitely a way. I also have to remember that engaging in consumer genetic testing is also something that is kind of like a privilege for people. Not everyone has access to buy these tests. Not everyone is curious also about their genetic background. And I think we just have to be mindful of those things.
Has anyone else encountered David, when you go out into the community, do people mention that they know about these tests, or like in a popular sense?
Consumer testing, not so much. No, not in the communities we see. I mean, just on the some of the community testing companies like 23andMe, they actually give a bit back and do fantastic research as well. So it's not just for those who've ticked the research box. We did a study mixing genes and health data with 23andMe data, and benefited both data sets. Got more than we would have done otherwise.
Yeah, I mean, even though they're cheaper and they're getting cheaper, they're still not cheap. They still are expensive. And I guess the idea of research or clinical trials is that people understand that they're not paid for tests. They're something that's a part of the process of science.
Yeah, and certainly in the UK, clinical trials are very much altruism based, or because you've got the disease or you know someone who's got the disease and so on. Yeah, there's a lot of altruism in our study as well.
So interesting. It's something I hadn't thought of actually, but it's very, very interesting to hear. We're coming to the end of our session. It's gone so quickly, and I just wanted to thank you all for your fascinating insights, but also for explaining really very complex science so clearly and succinctly and descriptively. And as we're approaching the end of the session, I wanted to just ask for your final thoughts in general, but maybe also to think about how long it'll take, do you think, before we, what we're thinking about is we don't have this diversity within genetic databases. How long before we see a noticeable, significant improvement to that? And any other thoughts you'd like to end with? Ananyo, would you like to go first?
Yes. So I think, a lot is happening. We have already heard about the UK Biobank, the Genomics England, the Genes and Health, all of us, million veterans. So, especially in some parts of the world, a lot of efforts are going on. but I think, I mean, this will happen in two layers. So the first is where it's easy, you capture the diaspora in US or UK. But then we have to remember that the diaspora is not there. It's neither representative, it's nor really, I mean, it's there because of some historical reasons and not because I mean, they went there by random selection or by representation. So I think after a point of time, and as if you remember the genetic diversity tree, so most of the groups which have been the major group, like some of the hunter-gatherer groups or the forager groups, they haven't migrated to anywhere in the world. So and this might be true for many South Asians group. So what we are seeing in diaspora is something which will be captured maybe in the next few years. You will see a lot of African, South Asian data that comes from diaspora, but then we will perhaps hit somewhere around the glass ceiling, and then genomics would need to move to these continents to really capture these populations. And I think so after that second phase, perhaps we will be where our databases will be really representative of the global genetic diversity.
That's so interesting. I know some places, I think India doesn't like to share population genetic data. I mean, there may be obstacles in some places more than others.
But even in India, the Indian government is running a huge project called Genome India, where they're sequencing 10,000 Indians from all diverse groups. So I mean, so every government everywhere in the world is also realizing that there is value in terms of what this brings to their population. So people are getting engaged, and that would really fill up the last bits of the puzzle, to say. Yeah.
We've also got data protection things. So individual level genomic data is personal data in Europe under GDPR. So you've got to be really careful with it and respect it and look after it. And you can put out summary counts of statistics and genotypes and so on. But certainly our approach for individual level data is that people come to us in a secure environment, and you don't take the data away, and care has to be taken also with sharing our genetic knowledge.
Thank you. And Janina, come to you finally.
Yeah, I was just, I think everyone has summed up this completely good. I think the only thing I would add kind of to David's point is I think there are going to be other barriers that we hadn't quite thought about that kind of stand in the way, and data privacy is definitely one of them. In the US, you see a lot of companies and initiatives making sure that they are GDRP compliant. And also, thinking about culture and respect and thinking about other things that play in part in other types of data that we would need in order to accomplish some of these goals, and if we will ever have access to those things, if we need access to those things. And so I think that it's a complicated question to answer. You hear a lot of people also talking about sequence at birth and thinking about how and if that data should be shared and how it should be used and so forth. And so there's a lot of discussion, but I think thinking about what are some of the kind of political aspects that we haven't quite talked about, and how could having too much data impact us as a society. And so I think there's a lot to gain from a medical perspective. I think there's a lot that we don't know and understand from a community personal perspective that I think we'll start to learn about in the coming years.
Someone once said to me, whenever you find a solution, think about the problems that the solutions bring, and in this case, think about the obstacles that there still are that we may not see. But I want to thank you all again. That's all we have time for tonight. But I feel very lucky. I think we're all very lucky to have such excellent and erudite and well-informed speakers this evening. I would like to mention that the next edition in the Open Talk series, and this series will take place on the 22nd of May. That's Wednesday, 22nd of May. And that's going to be a discussion that will bring together historians and railway workers to tell the stories of LGBTQ plus rail workers. And that coincides with the launch of the National Railway Museum's upcoming oral history project called People, Pride and Progress. And finally, if you'd like to support the Science Museum Group across all its 5 museums in the UK, they are all free to visit because their mission is to inspire the next generation of scientists, of technicians, engineers, mathematicians, and medics. You can find a link to donate in the description below. And so it just leaves me to thank our speakers. A heartfelt thanks to you all, Professor David Van Heel, Dr. Janina Jeff, and Dr. Ananyo Choudhury. And from me, a final thank you to all of you for staying with us and being a part of this discussion and watching this evening. A very good night to you. Bye-bye.
Thank you.
Thank you.